About 11 o'clock here, so let's get started. Thank you all for joining us today for this Lasergene Genomics Suite webinar. My name is Jackie Carville, and I will be coordinating the webinar today. I'm here with Matthew Kaiser, who will be going over some of the functionality of our Lasergene Genomics Suite, and then offering a live demo of our software showing a few different workflows for analyzing your microbial next-gen sequencing data. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and then select Send to Host. Alternatively, you can also follow us on Twitter and tweet questions and comments at our handle @DNASTAR Inc. I will then direct these questions to Matt to be answered for the whole group. If you need any assistance during the webinar, you can send a chat message, tweet, or email me at webinars@dnastar.com. And with that, I'll turn it over to Matt.
Thank you, Jackie. And I see that we have a number of folks still joining us here. I recognize a number of you. I know I've talked with you, a few of you, and worked through different projects. So hopefully we'll have lots of questions at the end. And hopefully I'll also cover a lot of the topics that you're interested in. So we'll go to my desktop here. And looks like we'll be sharing here right about now. Okay, so I thought I'd start today just with a little bit of an introduction to DNASTAR. I know some of you are already familiar with us, but we have some new folks as well. And so we'll just kind of give a brief introduction, and then I'd like to talk about some different workflows, some microbial workflows, and we'll look at the software. We'll set up some assemblies using a few different workflows, and then look at some of the analysis tools downstream. So DNASTAR is located in Madison, Wisconsin. Our sales team is here. Our developers are here. We have a very good support network. So if you purchase our software and have questions, we have the full staff on hand to help support the software and also use that customer feedback to really improve our development and make our software better with that feedback. A little bit on our history, our founder is Dr. Fred Blattner. Back in the mid-1990s, Dr. Blattner in his lab sequenced the E. coli K12 genome. So this was really a pioneering effort in genomics. And at that time, there was very limited options for software that could be used for looking at sequence data, assembling sequence data, doing the annotation work. And so they had developed this software in-house first to help with this project, and many of the tools that were developed in the lab were then commercialized and made available to DNASTAR customers. We've been in this business since 1984 and have really focused on solutions for life scientists and to provide powerful yet easy-to-use tools for them for Sanger data and next-gen sequencing data, and really give them research-grade software. Most of our software is desktop software, Windows and Macintosh. We do have our SeqMen engine that runs on a Linux cluster. And more recently, a little bit earlier this year, we had our first cloud offering, and so you can also use all of our software up on the cloud. So DNASTAR software really is research-grade software. It is the most cited software for bioinformatics in peer-reviewed journals, and you can see, dating all the way back to 1985, that we've had twice as many publications as the nearest competitor. So this truly is research-grade software, and what I hope to show you today is that it's powerful, yet it is intuitive and easy for you to use. So one of the first questions that we'll get, especially with next-gen sequence data, is how powerful is the software? And of course, these data sets are extremely large, many of them, and some folks think that you have to use a Linux cluster to handle the data, and you have to use open source tools. But what we've done is develop software that is extremely powerful and can handle even the largest data sets in a very, very reasonable amount of time. And so I'd like to show this benchmark, just our list of benchmarks from our website, to show some of the assembly times with these larger data sets. Of course, today we're going to talk mostly about microbial data sets, but you still benefit from having the fast assembler with smaller data sets as you start to batch them together, or if you're running oftentimes many assemblies in a day, it's nice to have one that finishes in a minute versus 30 minutes. So one of the benchmarks I like to show is the human genome assembly, and this is over 1 billion Illumina reads. It's deep coverage, 36x coverage. With our latest version of software, our assembly time is about 13 hours. And this assembly time is not just the mapping time, it's the time that it takes to align the reads to the genome, gap everything, do statistical SNP calling, generate all the coverage reports, the feature reports, and then build a BAM file with accessories so that you're ready to do the analysis right when the assembly is completed. So this time actually includes all the building of the output files. Smaller projects like E. coli are measured typically in minutes, and so in some cases it's under a minute to run the assembly. And other data sets like RNA-seq or smaller genomes, it might only be an hour or so to do the assembly. These projects that I'm showing with this list of benchmarks have all been completed on a reasonable desktop computer. You can purchase one from a vendor like Dell for under $3,000. If you can build your own computers and put some hardware components together, these computers can cost less than $2,000 to do even these largest assemblies. So very reasonable hardware. De novo assembly uses a little bit different algorithm. De novo assemblies are RAM dependent. So our assembler SeqMan engine is very accurate, produces very accurate and precise contigs, but it is RAM dependent, and so we generally limit our de novo assembly to microbes or eukaryotic transcriptomes. This might include some smaller eukaryotic genomes. Generally, under a gigabase genome or under even 100 megabases is a reasonable size limit. The metrics that we measure, certainly we look at the time, but that's probably not the most important metric. Really what we're interested in is how accurate are the contigs. That's the number one metric, and also how large are some of the contigs. And so some of the typical results that we'll get is with E. coli genomes that are more repetitive. We may get a contig on 50 of 100,000 base pairs, and it'll take 15 to 30 minutes to do the assembly. Again, the time is highly variable depending on how much data you have, how deep your coverage is. Transcriptomes, again, can vary tremendously in size, but we produce very, very accurate results for transcriptomes, and it can be several hours to an overnight assembly. Less repetitive genomes like Bacillus, and this is actually a genome that's up on the cloud on Illumina's base space where we have SeqMan engine, and that can produce tremendous assemblies, contig N50s of well over 200 kilobases. And so our de novo assembler, again, it's very, very accurate. It's well suited for assembling microbial genomes. You want accuracy over speed or contig size, and that's something we can look into some more detail. So the DNASTAR software really supports as many next-gen workflows as is possible. And so we try to cover all the bases so that if you have our software and you have researchers doing a variety of different projects, that we can support all of those workflows. And so that includes things like genome resequencing. That could be human genome resequencing or a non-model organism. Targeted resequencing and exome sequencing. Targeted resequencing oftentimes is a set of amplicon sequences, and it's a very different kind of geometry to the assembly. Targeted resequencing is oftentimes thousands of sequences deep. You're sequencing something deeply to look for rare variants. So our algorithm can adapt to that type of a project. RNA-seq, aligning sequencing RNA, and then aligning that RNA back to a genome or a set of reference sequences, and then doing things like looking at quantitation of expression or looking at exon usage patterns. So we have software to do that. What we'll look at today, I have bolded here, we're going to look at some microbial metagenomic workflows. Some of these are new workflows in our software, so I know many of you have our software. You may not be familiar with some of these new workflows, so we'll look at that today. De novo transcriptome, automated bacterial genome closure. We'll touch on that a little bit today as well. Of course, I've had a webinar late last year where we went into this workflow in detail, but we'll integrate this workflow a bit with the microbial metagenomic workflow. And then, of course, we can do de novo genome. Another new workflow is viral genome and host integration. Again, this is a new workflow for looking for retroviral integration sites in the host genome. So lots of different workflows. We'll look at the interface where we can see where we make the decisions on which workflow to choose. And so I think where we'll start is the microbial metagenomic workflows. And this is kind of a bit of a hodgepodge of different things that you can do, and we'll try to look at examples of each of these. One of them is, it depends on what it is that you've sequenced, will determine what kind of a workflow that you want to do. So one of the applications is to take metagenomic sequence data. Maybe you sequenced all the organisms from a soil sample or a seawater sample. We've seen different data sets from all different kinds of environments, and you want to find out what the composition is. And so we can use our metagenomic sorter to take the sequence data and then align that sequence data to a database, either a set of reference genomes or it could be one of the 16S databases that you've downloaded. So that's one workflow where the assembler takes complex data and parses it to a variety of different database templates. Another approach with metagenomic data is a de novo assembly. So if you have microbes that you've sequenced, and many of them are novel and maybe not represented well in the bacterial genome databases, or you could take the leftover sequences that don't align to the database sequences. We can collect those and then do a de novo assembly with those. And that's a way to discover new organisms, cluster them into contigs, blast them, identify them, annotate them. So again, a whole different kind of a workflow for looking for more novel genomes. There's also a kind of a new feature in our software, which is the removal of host genome contaminant sequences. And then we can follow that automatically by either a sort, a templated assembly to the reference genomes, or a de novo assembly. And in the past, that required multiple steps. You'd have to do one assembly to a host genome and then collect the unaligned reads and then set up another assembly and then pick your workflow. This automates that process to make it quite a bit more streamlined. So we'll take a look at that as well. And then finally, there's another workflow or another kind of a goal, and that may be if you have a sequence strain rather than a complex strain, just a single isolate, and your goal is to sequence that genome. And so one of the first decisions that you'll have is there a good reference genome for my clinical strain? And what you will want to do then is try to find, if you don't know what the best reference genome is, or you want to confirm what you think might be the best reference genome, you can align that data again to a microbial database or a partial database and determine what the best strain or strains are to act as a reference sequence. So it's all different kind of varieties of this, working with microbes that integrate with different workflows. So at this point, we'll just jump out and take a look at the software. And so we'll start with SeqMan Engine. And, that's not quite. And we'll start with a de novo assembly. I'm just going to back up here a little bit. So we'll have a new assembly project. And so what we're looking at here is the SeqMan Engine. We call it a wizard. And it's a wizard in the sense that this allows us to set up a variety of different project types. And whether we're working with human sequences or microbial sequences or de novo or templated, ideally we have one assembler like Engine that can accommodate all the workflows and with minimal user input allow us to be successful with wide-ranging types of assemblies, which provides a nice advantage over, say, open source, where you might have to learn a separate Linux-based software for each type of sequencing platform, each type of assembly. So SeqMan Engine can really integrate these assemblies for you and streamline that process. So what this wizard is doing is, as we make selections, create new project, then we choose a project type. The project type then really determines our workflow. So I've selected a metagenomic or a 16S RNA assembly workflow. And as you make these selections, it actually changes the following screens and will set optimal parameters for this particular workflow. So for instance, if I pick metagenomic workflow, I've got some different choices here. I have a templated assembly. I have a templated assembly with host removal, a de novo assembly, or a de novo assembly with host removal. I can contrast that with my genome assembly option. And I have different choices here, a de novo, a templated, a special templated with gap closure that we'll look at towards the end of today's webinar, and then actually a heritage workflow. And so again, as we make these choices, that changes what we see in the following screen. So in this particular example, we'll do a metagenomic assembly, a de novo assembly, and we'll incorporate the host removal. And so there's a lot of different examples of when we want to do these types of projects. I've typed this in already, metagenomic de novo with host removal. We'll save the project out as a SeqMan Pro editable file. If our data set is large, above 10 million reads, we can also save as a BAM format, which does not have a read limit, but it's not editable. But it allows us to look at really big de novo assemblies. So we'll have a couple options there. Then a new point in the SeqMan Engine Wizard now is to input the host files. And in this case, we could add FASTA files. We could add GenBank files for the chromosome sequences. If your possible host contamination is one of the model organisms, like human, in this case, this is a human gut bacterial sample that was, and so there's plenty of human DNA present. And for many different types of assemblies, especially de novo assemblies, it's essential to remove the host contaminants. If I do a de novo assembly with a lot of host genomic sequence data present, what happens is that host genomic data is present at very low coverage, less than 1x in most cases, and that will cause, and usually the repetitive elements will be overrepresented from the host genome. And so what will happen is, in memory, you'll get thousands of little tiny contigs that are usually some repetitive element in the host eukaryotic genome. And that will foul up the assembly. It will use up a lot of RAM. It will take a lot longer. It might not finish if there's too much of the host DNA present. So there's a lot of good reasons to remove this host DNA. So we give this option up front. We can load in a template. Then we can select our sequence files. This is the point where we can specify the sequencing platform. We distinguish between the short and the long Illumina reads. These days, most people have Illumina reads greater than 50 base pairs. In a de novo assembly, length of read matters quite a bit. And so you get a more stringent assembly. It's easier for the assembler the longer the reads are. So you'll certainly have 100 base pair Illumina reads. Hopefully you have MySeq reads or something that are 200 or more. Or in some cases we have 454 reads or ion torrent reads that are longer and will assemble well. So I can pick my technology. This really starts to set the optimal parameters for that sequencing technology. There are different optimal parameters. Each technology has its own quirks. It has its own error rates, its own quality scores, its own way to handle pair data. And so we tell the assembler what type of data we have. It sets optimal defaults for that data. I can then add my sequence data. In this case, I've added 4 FASTQ files. So FASTQ is the typical input format for SeqMan Engine. Occasionally we have SFF files from 454 or ion torrent customers, but in general, FASTQ has been the predominant input sequence data type for our assembler. Then we can pick some read options and some trimming options. Again, this is for the de novo assembly part. Oftentimes we want to do a quality trim, and that's on by default. So again, de novo assembly requires very clean data. If you have linkers, primers, adapters, or low-quality ends on the sequences, that will tend to truncate contigs, and contigs will end prematurely with that low-quality data. And so we'll do a quality trim. It's also possible to do a vector or adapter scan. And this is, if we know that there's some vector or adapter or linkers, we can specify and then go browse to a FASTA file that contains those sequences. SeqMan Engine is an excellent trimmer, and I won't go into these in detail, but we have all different trimming options: fixed end trimming, quality trimming, vector scans, So it's a very, very effective trimmer. We use this also for de novo genome assembly. So if you sequenced your microbes and you don't have a good reference sequence, Seqman Engine is excellent for trimming the data so it's clean and then creating very high quality contigs. We have some more assembly options here. I won't go through all these in great detail, but minimum match percentage is the most important of all the options, and this really sets the stringency of the assembly. This is a metagenomic assembly, so my expectation is that I'm going to have multiple bacterial species. This is human gut, and some of them might be pretty similar to each other. And in fact, they may have conserved regions that will cluster together into contigs. And so I'll want to oftentimes increase the stringency, and this is the minimum match, and that's the percent that the read must match the consensus sequence in order to align. And so by increasing this number, I can make a more stringent assembly. It has the same effect in a de novo genome assembly. I can make it more stringent, make more accurate contigs. They may be smaller contigs, but again, accuracy usually is more important than contig size. So if you're going to change one parameter in our software, I recommend minimum match percentage and start with the default, go up by 5%, see what your results look like. We can also remove the smallest contigs after assembly. The reason for this, again, is if we have some remnants of the host genome, we could have 10,000 little tiny contigs that don't get removed effectively. Or we have some small junk sequences, essentially. And that will make the project more sluggish, slower to open, larger. So we like to cull out the smaller contigs from the project. And then we're ready to begin. So de novo assemblies actually have more options. We'll look at a templated next, and you'll see that it's actually a simpler interface. De novo assemblies require a little bit more thought about how I'm going to trim my sequences and treat the sequences. What happens here is that we have the wizard generates a script that we save out. And this script file for advanced users can be useful. Some users prefer to use Engine from the command prompt, and they might want higher throughput options to take multiple scripts, thread them together, and run multiple assemblies concurrently. So there's different advantages. Our QA department certainly uses scripts. They run, dozens and dozens of assemblies a night with a master script to do that. So you can see all the parameters here for the script. If you're trying out a demo of our software now and you have questions or technical support, oftentimes I say, send me the script file so I understand exactly what kind of assembly you've set up and what parameters you're using. When you click Assemble, we'll see a log, and this log gives us information about what's happening during the assembly. Right now it's going through all the human chromosomes and loading them up, and it just loaded all the sequence data, so I can see information in the log. And again, if there's a problem during assembly, we can export the log and send that to DNASTAR as a text file, and we can troubleshoot your particular project using this log. So it's very, very useful. And you can see it goes through this. This is a small project with a so it assembles in a few minutes, but I'll stop it before it gets too far. I'm just going to go to the task manager, and we'll see that this alignment to the human genome launches our XNG program. This is small enough that it's not going to tap into multiple cores, but if you have larger projects that you're aligning to a template, Seqman Engine will use multiple cores. It'll use a chunk of RAM. For your de novo assemblies, it'll use just a small part of one core and mostly RAM for the assembly. So it's just something to keep in mind, especially if you're working with microbial genomes and you're used to just using RAM for your de novo assemblies. If you start doing metagenomic sorting, or if you start doing host removal, then that's actually a templated assembly that we're doing. Then you have to pay attention to things like disk space and having enough space for temporary files. So it's a little bit different hardware requirement. So this will finish in a few minutes, and there will be a button here that says Launch in Seqman Pro. Actually, there will be a report first that tells us what happened, how many sequences aligned, and some other metrics. Then we'll click Next, and then we can launch the program or the assembly automatically in Seqman Pro. So Seqman Engine is the assembler. It's 64-bit, but the output file can be analyzed in our Seqman Pro software, which is part of our our core package. Many of you have access to SeqMan Pro already. It's been used, out in the real world for many, many years. So the idea here is that you can run assemblies with our new software, but the output can be analyzed in our standard software. So I'm going to stop at this point.
Hi, Matt. Before we move on to the next assembly project, we do have a couple questions that came in. So the first couple questions, people have noticed that there was a paired end read file input section in one of the screens on NGen. Can people add in paired end reads with single end reads in one assembly?
Oh, yeah, absolutely. So if you have fragment data, single end reads, you can add that along with the pair. You can also add multiple pair insert libraries. So you might have paired end data of a couple hundred base pairs, or mate pair data with several thousand base pair inserts. You can mix and match those together.
Great. Kind of related to that paired end reads, what do you recommend for paired end library sizes for bacterial genomes?
There's the ideal answer to that, and then there's the practical answer, I guess. Ideally, you would have pair inserts that span the largest repetitive elements that are present in the genome. So for E. coli, those largest elements typically are ribosomal operons, but they could be things like phages that are inserted. And then, of course, there's many different IS elements, transposable elements. And so what works best, ideally, is to have mate pairs maybe 10 kb, and then have some paired end data that's only 500 base pairs. And so the reason for that is that in a de novo assembly, contigs typically end where they hit an area where the assembler says, well, this read goes in 5 different places. So I'm not going to place the read in this repeat because it could falsely join contigs together. So that's what leaves the gap in the contigs. If that gap is due to a ribosomal operon that's 8 kb in length, in order to put those contigs or order them in the scaffolds, you need a mate pair that can span 8 kb. So a 10 kb mate pair library would allow you to scaffold those two contigs next to each other. So that's what's ideal, is to have some mate pair. What's practical might not be to have the mate pair. Mate pair can be expensive. It can be difficult for some folks to generate good mate pair libraries. So it's probably more typical that we see paired end libraries with 500 base pair inserts. What that means is that you won't be able to order contigs through those larger repetitive elements. So.
Great. A couple other questions before we move on. If we have reads from 2 assembly platforms, let's say 454 and Illumina, can we use these reads together at the same time for an assembly?
You can't use them up front together to get the best result. So what that means is, yeah, you could feed them both in. The problem is that the assembly parameters are different for 454 and Illumina, or Ion Torrent and Illumina. And so, but it is advantageous to have both platforms in many cases. So there are data sets that we've used and many of our customers have used where you can take the 454 reads, if they're titanium data with mate pair, long reads, large inserts, assemble the whole genome with 454 data, and you get the largest contigs that you're going to get. And you can scaffold it into big scaffolds. Then you can take the Illumina data and align it to the contig consensus sequences. The Illumina data does a couple different things. One, it removes all the homopolymeric errors that are present in 454 data. So it does a massive error correction. The second thing it does is it adds depth across all the contigs. So you can confirm the accuracy of all the contigs generated with the 454 data and then edit them where there are problems. So it's not assembling it up front at the same time. It's doing an iterative assembly, but it takes advantage of what both platforms have to offer.
Sounds great. Thanks, Matt.
So I've opened our metagenomic de novo project, and I'll just show just a couple things here. I opened a project report. So under project report, and we get this report that's, again, very useful for troubleshooting, and it gives us some metrics. It tells us what version we used, how long it took to assemble, how many contigs there are, things like assembled sequences, unassembled. We can see here we just have 1,392 out of 161,000 sequences. There's a lot of human contaminant in this sample. So we're very glad that we did host removal on this particular sample. We get a contig N50 and then a breakdown of everything else, of the script and sequence lengths. So that's the project report. Then we get a list of contigs, and again, this is a human gut metagenome assembly. Most of the data was human data that was present, and so we get some smaller contigs, and what we can do then is we can double click on a contig, and you can go and actually look at the assembly. You'll see that a consensus is generated for each of these contigs, so I could export the consensus. I can also do things like annotate it, create a feature for this contig. I can rename it. I can also blast it. So if this is a metagenome and I want to know what was in this sample, or if it's a leftover from a templated metagenomic assembly, I can do a batch blast. In this case, I highlighted a number of the contigs and blasted them under net search, and we get this blast window that is really useful for these kind of searches. And what it's showing me is, scores and e-values, but then this query coverage is what I like the most, and it tells me which contig was blasted, the range of the contig, what percent of the contig matched the database hit. So I can distinguish between 100% blast hits that are only 20 base pairs versus the entire sequence. So it gives me a way to differentiate between these high score blast hits to look at the blast hits that cover large portions. And so I can then see my query, my database, where it hit the database sequence, and then I get a description of that database sequence. I can sort by query coverage, so here's contig 14, and I can see contig 14. I can see all the organisms that it's hitting. And I know that Bacteroides is one that's present in the gut at high levels. I can click on one of the blast hits, and it will give me the pairwise alignment and the percent and the identity. So it's a really nice blast window for doing a rolling batch blast of de novo assembled contigs. And so that's a nice way, and again, Engine is a very stringent de novo assembler. We get very high accurate contigs, and we can send these out to do annotations or do annotations within SeqMan. So that's one of the workflows. I'd like to show another workflow, and we'll go. And this is an instance where we've got a metagenomic mix of microbial sequences, and we want to align it to, we want to see what's in that sample. We don't know for sure, and we're going to align it to the largest collection of bacterial genome sequences that we can find. And so I can, again, I'll just back up a couple steps. So I can create a new project. Again, pick the metagenomic workflow and select templated assembly with host removal. And I'm going to call this bacterial genome database. It'll be a .assembly output. And here's where we're going to have some, you'll notice the interface has changed a bit now. It's templated. Now it's going to ask me something that de novo assemblies do not ask. It's asking, where do I put the temporary files? Temporary files are used for our reference-guided assembler in lieu of RAM, so that as data sets get very, very large, you can use more disk space. So it allows us to do the really large data sets very quickly by writing some of these files to disk. We can input the host files. Again, we're going to use the human genome for this sample. And then we can input the biome genomes. And so these genomes can be your own collection of FASTA files. You might have a focused set of reference sequences that you're interested in. You can also download from NCBI or from a variety of sources of different genomes. This particular folder here that I'm loading, I'll just take a look here, is a microbial genome database. And this was downloaded from NCBI, and it's a little tricky sometimes. So part of the challenge to doing this type of an alignment is getting the reference genomes that you want. And so we download these from NCBI, and we can share these with customers if you're interested. And we did a little modification to this database. So I'm going to use my universal viewer to take a peek inside. And databases will oftentimes have a FASTA format and then use their index number first, and then some kind of a delimiter like a vertical slash, sometimes it's just a space, and then the species name, and then some other stuff. And so that can be variable depending on the database. So what we did is we just went in and swapped out that ID number and put the species name right up front, so we can in the FASTA header. So that's typically what's pulled throughout our software so that you can identify what these different sequences are. So we made a slight modification there. And so the alignment in this case is going to be to all of these are each folder alphabetically organized database. I believe it contains around 3,500 bacterial genomes. And so we're loading the whole folder in, and we're going to align our sequence data then, again, just these FASTQ files, to all 3,500 bacterial genomes. And now this is a templated assembly. It's a little bit simpler, some fewer options here, but we still have our minimum match percentage. And really, we want to set a fairly high stringency. And what I mean by that is the default, if that's 85, we want to go up from there and try 90, try 95. If we go too low, what will happen is you'll have too many reads that align to too many different templates equally well, especially if your sample contains many species that are similar. So as we increase this stringency, we parse the data more accurately and more specifically to the different templates. Again, SeqMan Engine is a really, really fast assembler. If you've got enough disk space, it'll crunch through this data and align all those templates in just a couple of hours. So it's very, very fast. It gets faster after you've used those templates a second time as well. Once it breaks down all the MERS in those templates, that file sticks around. So if I reuse it, it doesn't have to recompute everything. So the second time on that I use that template, we get even a faster assembly. There's some more options here. One of the keys here is the reads that map to more than one template. The primary difference between a metagenomics sort and a reference guided genome assembly is what you do with reads that map to more than one place equally well. When you have a metagenomic alignment, there's many strains that are very, very similar to each other. So you would expect reads, even in unique areas, to map to all of those templates equally well. With a metagenomic alignment, we allow the aligner to place all of the reads. We don't force it to make a decision or assign a partial read. It's going to place all of them. There's some. We can also pick genome ploidy. That's going to be for SNP calling. This is a population. And again, we have a script that's ready to begin. Now when I click Assemble, it's going to try to process all those bacterial genome templates. So it's a pretty big load of sequences, not terribly huge, but it's sizable. So now when I go to my task manager and I look at XNG, it's at 99% of my CPU. So the webinar like this might start getting scratchy, and we might start losing audio if I go too long. What I'm showing is that the software is really optimized for maximal performance. It's going to use for these larger templated assemblies, it's designed to use every resource on your computer to the max to get the fastest possible assembly time. It'll do this. I've got 16 gigs of RAM. It's at about 9 right now. For the first 15 minutes or so, it'll max out. Then it'll settle down later, and I can check email and do some light duty on this computer at the same time. But I'm going to stop this before our webinar stops. So let's see here. We'll do that, and that shows you also in what the error log looks like. So if you're having hardware problems or the assembly terminates, you'll get a red message that it wasn't successful. Again, you export the logs, send that to DNASTAR, and we can help you out with it. And we'll look at the output here. Oops. And I believe it's this file. Nope, I guessed wrong. Okay, so here's the assembly now of our human metagenomic data to the bacterial genome databases. And what we get, there's a couple different output options. One is the SeqMan project file. And I can go and look at the project file, and what I'm looking at now is the config list. And because we restructured the FASTA files, the species name now will show up as the name of the config. And I can see how many sequences aligned, and I can see the length of the template. So by sorting by sequences, I get a pretty good feel for what the most abundant bug was in this data set. So it's just kind of a quick way to see, what is most abundant, what's present in this sample. Of course, there's the other views. I can look at a strategy view to see where the reads are aligning. And you can see it's pretty evenly distributed throughout this genome. Not a lot of coverage. It's a pretty small data set. And then I also get some project statistics. And this will show me number of sequences, median coverage on the project. Now, you might not even have to look at the project. You might just want to list. So there is another output report. So for the really big metagenomic sorts, if I sort to 1,000,000 16S RNA sequences, and I've got 500,000 hits, there's a point where that becomes unwieldy to open and load a project. It might take so long to load the BAM files that I'd rather just look at an Excel table that gives me the answers. So we do provide another output, and this is new with our software as of this year. And I'm going to show you where the file is here. It's so in your project output, there will be a file called Summary of Results, and so this will be saved out for these projects. And we can open this file in Excel, and in our Excel table, then it'll just show us. And I prettied this up a little bit. I'll have to admit I deleted a couple columns just to show the ones that I'm most interested in. But we have the template title. So here's the name of the organism, the length, the sequence count, the number of SNPs that were detected, and the percent of the template that was covered. And so again, this last column is really an important one because of the way metagenomic data will sort. You might get a pileup on some of these templates just in one little area, some conserved element. And in that case, you might get a fairly high seq count, but a pretty low coverage count on the template. So you need to be able to distinguish between lots of reads, evenly distributed, versus reads that are piled up. So template coverage percent confirms that this Bacteroides is the most abundant bug in this particular sample. And so again, that's a tad delimited report that we generate. It goes out into the project report. And so if I have really big projects, I may go here first, figure out which organism is present, and then proceed down my path. And that kind of leads me to the last workflow, which really integrates with our automated bacterial genome workflow. And one of the main questions with the bacterial genome workflow is what is our best template that's available? And I have many, many customers that start projects and don't know what the best template is. So you spend a lot of time either de novo assembling or using the wrong template and spending a lot of time when you could use this tool to find the best template for you. So one thing that we could do is set up our project. I'll just kind of go back to the metagenomic workflow, and instead of aligning to an entire bacterial genome database, I could go in and take a smaller piece. So if I know I have an E. coli strain, for instance, I can go into NCBI and get all the E. coli genome sequences. And so I might usually know what your strain is related to, but if I have a situation where I've got an E. coli strain, but I don't know which one it's closest to, I can do my metagenomic sort to this smaller subset. And then once I identify through the template coverage and looking at the assemblies, I can pick my best reference then and pick the one that looks to be the closest. And so that kind of leads us then into another workflow, and I'll just jump over here to...
Really quick, before we start this next workflow, Matt, one quick question was on some of the technical requirements for doing these metagenomic projects in SeqMan Engine.
Yeah, that's a great question, and the answer comes down to how many templates you'll be aligning to. If you're going to align to all 3,500 to 4,000 microbial genomes, you need enough disk space to process all the templates. And I don't have an exact number for you, but I can give you an approximate number here. Let's go to, so I'm just going to jump to our website here. And we'll go to Technical Specs, SeqMan Engine. And, oops, I missed it. Support, technical specs. So on our website, we have a number of support areas, and under the technical specs, we have specs for reference guided assemblies. We don't have a metagenomic spec up here, but I know that when we put all the microbial genome templates together, I believe it's about 1.5 gigabase in size. So it's roughly, we'll call it roughly the size of a human genome. So we do human genome alignments, human exomes. The disk space is about 2 terabytes is what we would recommend. So if you're going to align to the whole microbial genome database, I would recommend a 2 terabyte drive. It can be an external drive connected by eSATA or USB 3 or an internal drive. That will give you the best performance. And so the core computer can be a pretty standard 16 gigabyte RAM, quad core, but how the size of your data sets really determines how much disk space you need. So if you're not going to be aligning to a whole database, a terabyte of space is more than enough.
Great, thanks. And then one last question before we move on. Someone had noticed that in the SeqMan Engine Wizard that there was the option for 16S data as well for these metagenomic assemblies. Are the workflows similar if you're using that 16S data?
Yeah, they're the same. So I'm using this example interchangeably with 16S. The 16S databases, we've downloaded them from Silva and RDP as FASTA files, and again, depending on how the FASTA file header looks, you may have to do a little editing to get the species name correctly in the FASTA header. The primary challenge of 16S is that there's just more templates, and there's over 1.2 million or something like that on RDP. We've downloaded it here with customers. We can do the alignments to 1 million templates. Depending on your data set, though, if it's a highly diverse metagenomic sample, it might align to 500,000 of those, and that creates a lot of BAM files. And actually too many to really open. It takes too long to open all of them. That's where the tabular output, having the Excel sheet that just shows the summary of the results, that's the preferred output for a 16S data set.
Great. Thank you, Matt.
You're welcome. So one last thing. Now this is the automated bacterial genome closure. So this is a workflow that, again, back in December, I had a whole webinar devoted just to this workflow. So again, on our website, under the support, under webinars, I focused just on these workflows. At the time, though, we didn't have the metagenomic, kind of the genome picker idea to lead into this workflow. So that's what I focused on today. How do you get that, find that best genome? Once you have a genome that looks to be close, you can use our workflow to really guide the assembly of your sequence strain. And so the question always is, should I use the best reference? Should I use a de novo assembly? And usually you don't have an answer until you just try both. With a de novo assembly, you're going to get contigs, and the number varies highly depending on what it is that you sequenced. A repetitive genome, like some of the E. coli strains that have lots of IS elements in them, you might get, you could have 300 contigs. Now, of course, many of these are small that you won't have to deal with. It might be the top 50 contigs that you need to put together, or the top 100 to get your genome. But it's a lot of contigs. If you've got good mate pair data, you can scaffold most of it. If you have all paired end data with small inserts, you've got a lot of manual scaffolding that you would have to do. If you have a novel genome that is not represented in the database very well, it can be a pretty challenging approach. So de novo assembly is one option. Just look at the number of contigs and see how many gaps do I have to close manually to complete this project. Then there's also the alignment to a reference, which is kind of the old workflow. I take the best reference, I align it, and I look and I look at the assembly, and if I have novel regions in my sequence strain, in this case we've sequenced MG1655, K12 strain, and we're using a closely related strain, MC1061. At areas where there's novel elements, we get gaps in coverage. In the old workflow, I would look at that and say, okay, I have to break my reference here, split it apart, scaffold it, remove the reference sequence, and then manually resolve each one of these gaps. And with these two strains, it's about 30 different gaps that are present. And so I'd have to manually go in and do about 30. If I figure it takes me a couple hours per gap, we're looking at least a couple of weeks to do this project and to finish it up at a high level of confidence. The new workflow automatically recognizes these areas and says, okay, there's a disruption in the alignment pattern here, something's going on, split the reference, use pair data to drop pairs into this gap, anchor the unique portion of the read in the flanking regions, build contigs using the pair data, and then look for overlap. And it looks for deletions and insertions that way or any other anomaly in the assembly. And then close things together. And a typical result when we have a reference that's fairly closely related, and you're talking from 10 to maybe 300 structural variations differing between the strains, we can close between 80 and 90% of those gaps automatically. And so as a time saver, that's, reducing my project from a couple of weeks to maybe a day or two. So it's really a nice tool to speed up the process, a very difficult process for many people of closing gaps. So the way that it works is that it finds structural variations, it splits apart the reference, removes the reference bias, it excises reference sequence into deletion areas, merges split reads. So there's a number of steps, and it maintains the contig order throughout the process. The insertion sites are recognized by trim points. So if your sequence strain has an insertion, the reads that extend into the insertion will get trimmed back as they're aligned to the reference. So it has this kind of a phenotype in the alignment, pretty clean. And when it's homozygous, it's very clean and obvious. There are other structural elements that aren't as obvious, but this is just a very clear example. Deletions are detected by two different metrics. One are reads that map to either side of the deletion and are split by our assembler. And I'm showing a strategy view here where it's colored pink, and there's a key up here in the left corner where I can control what's visible down below, and I'm just showing the split reads. The coverage map shows this great drop-off in coverage. And so this is obviously a deletion. They're not always this clean. Sometimes there is some random alignment if it's a repetitive element, so you get 50% drop-off, but you still get the split read, so you still know there's a deletion there. So we have very nice algorithms for detecting these areas, and not quite there yet. I'm going to jump out here before I say thank you. Let's go to, so I do want to show a couple of quick things here. So for this workflow, we choose genome assembly, and it's reference-guided assembly with gap closure. So this is the third of the genome kind of workflows, and we assign a name, again, temporary files. We supply the template, the single template that we're going to use. And in this case, we load pair data. We need the paired data to fill in the novel insertion sequences. And so we're going to load in pair data. We could add single-end data as well. And this is a haploid, set our stringencies, and we're ready to go. And most of this will be automated then. So it'll do the first round of assembly, it will split, and we get a couple of different output options, and it'll prompt you and show what's happening. And see, that's the middle. And what we'll get is a project then after it's done the splitting that looks something like this. And what's happened here is our reference is MC1061, and we've got a scaffold that was automatically created, and you can see that there's 5 contigs here. And so what that means is that even though there's 31 structural variations, all but actually 4 of them were able to be automatically resolved. And what we're left with then are these large contigs. You can see there are hundreds of thousands of base pairs. We have the position information, and this position information relates to the structural variation report. Oh, it doesn't want to come up for me. And at 62,937, I know there's a large insertion, and I know at 122,6,241 is another, I think, a ribosomal element. And so what's unresolved here are just the larger insertions that we didn't have quite enough mate pair data to make a contig to fill in that whole area. And so our starting point now, instead of just a de novo assembly with 100 contigs or a reference assembly with 30 structural variants, our starting point is much closer to the end point, and that's a scaffold with just a few large contigs in it. So again, it's a great workflow. It gets you much closer to the end point. Within an hour, you're at this point versus a couple of weeks of effort. So that's we're about out of time. I imagine there's going to be a few more questions, and I will show you before I'm going to go back to our website. I just want to show under the support and webinars. You can see some of the past webinars on a similar topic. So if we didn't cover everything that you wanted to see, I covered back in January genome assembly. This is microbial genome assembly and gap closure. So it's using the gap closure tools. And then also this automated workflow back in December. I spent the whole webinar showing the details of this automated gap closure workflow. So those are a couple of webinars that you might want to watch. And then there's also some specific Illumina and Ion Torrent webinars that also do some of the microbial work as well. So with that, I'd like to thank you for joining me for this hour. I'll take any questions. If we don't get to you now, I can respond to you through email later today. Thank you.
Great, Matt. Well, one quick question before we sign off for the day is how related should the reference be for you to use it in this automated genome closure workflow?
That's a great question. That's the million-dollar question. And the best that I can advise is do a de novo assembly and see how many gaps you have to close, and use your best reference and see at the end point how many contigs and scaffolds do I have. And if one of them has many fewer contigs, that's the route to go. Now the limit or how close the reference strain has to be, you pretty much have to run the assembly to see. I hesitate to say how any kind of a number for how conserved it might be, but I think a general rule of thumb is that it has to be under about 300 structural variations that are in the SeqMan report. If it's under 300, you generally will get that will be an advantage over a straight up de novo assembly. So if it's more than 300, you're probably better off just going de novo and not letting the reference bias contig formation.
Great, thanks, Matt. Well, it looks like we're about out of time for today, so we actually received a lot of questions, and if we weren't able to get to your question, we will definitely follow up with you via email after this webinar. In addition, if you think of any other questions later, you can email those to me at webinars@dnastar.com or tweet at us at DNASTAR Inc. Also, feel free to contact me with ideas for any future webinar topics. So thank you again for joining us today. We hope you found this helpful. I'll show you right here. We actually have a large collection of videos on our website, so feel free to check those out at any time. In addition, you can check out our webinars page that Matt was showing you to view the recording of today's webinar, which will be posted within 24 hours, watch any of our previous webinars, or look for new webinar announcements. So thank you again for joining us today. We hope to see you again soon. Thanks a lot, Matt.
Thank you.